Hey, hey, 1, 2, 2. 2, 2, 1, 2. Hey, hey, sure. 1, 2. 1, 2. Hey, hey, 1, 2, 2. Hey, hey, sure. 1, 2. Check. 1, 2. One, two, hey, hey, sure. 1, 2, 2, 2, hey, hey, sure. 1, 2, 2, 2, hey, hey, 1, 2, 2, 2. Hey, hey, sure. 1, 2, 2, 2, hey, hey, 1, 2. Hey, hey, 1, 2, 2, 2.
Oh, okay, wonderful.
Just going to check on the slides.
All right, great. Are you on the panel that's happening?
I am.
Oh, okay, cool.
I think we'll all be up there for the whole thing.
Okay, great. And then when you're done checking that, come back here and I'll get a microphone on you.
Sure.
Hey, hey, 1, 2, 2, 2, sure. 1, 2, check. Hey, hey, 1, 2, 2, 2, hey, hey, 1, 2. One, two, hey, hey, sure. 1, 2, 2, 2, hey, hey. 1, 2, 2, hey, hey, sure. 1, 2, 2, 2, sure, hey, hey. 1, 2, 2, 2, sibilance, 1, 2, 2, 2, check, 1, 2, hey, hey. Hey, hey, sure, 1, 2, 2, 2. Hey, hey, 1, 2. 1, 2, 2, hey, 1, 2, 2, 2, sure. 1, 2, hey, hey, 1, 2. 1, 2, hey, hey, 1, 2. 1, 2, 2, 2. Sure, 1, 2. Hey, hey, 1, 2. be on. There you go.
Testing 1, 2, 3. Testing, testing. You got to get mic'd up too. How you been, man? Is today your birthday?
Yes.
Happy birthday.
Thank you.
Sue got a very nice award.
Oh, really?
Oh, yes.
So you get lunch at least?
Yeah, it's good. Little, Mediterranean food.
Constellation prize. So, how's it going, man?
Good. You bring your jacket to work today?
Yes.
I just checked all the slides. They work fine.
They were fine. I mean, like,
I don't remember the script though. I gotta look at the.
do you have it? I don't have
I'm
it.
gonna try to find what Sue said. I'm hoping it'll just be obvious.
I just know that where to pick up from and then where to leave it off for you.
So you doing something fun with your family this evening?
I got my parents
Cool.
in town, so it's a little tough to get. I would really love to get a beer, but that would be a lot tough.
Rain check?
Yeah. So, what's up with you, man?
Not a lot. I mean, staying busy.
Not a lot. I find that hard to believe.
No, I had a lot. I got some deadlines and stuff coming up, but I applied for promotion. So I got everything in.
I heard you got an hour for.
Not an R, but I got a FNIH project. It's $5 million. So it's basically the same thing.
I don't know, maybe twice as good.
Yeah, but it's me and I'm co-PI, so I get half. How about that? So it's about, and then my trials.
Don't focus on that, okay?
Everything's going well. How's your stuff? I swear that Sue said something.
They don't promote you.
Say what?
They don't promote you.
Okay. I'll ask Carri for a job. Scott Reeder is radiology.
I'm trying to put like a couple things in humans.
John Ingalls scanned their first manganese 51 patient.
Anything. One is I think that they're just going for something different.
Oh, really?
So from pain kind of thing.
They were looking for like joints or heart or whatever.
Yeah. And then I showed the pain. It's like,
I saw there was a good like radiology story about Exendin. I wonder if if Exendin shows promise, we could do like a head-to-head.
I know about. I mean, that's not all bad, but a pretty good amount about sending. Like one with the Dutch.
Yeah.
No, Stevie.
Stevie Graves.
Rafael Diaz.
Nice to meet you.
My partner in crime here.
Aha.
To get stuff.
This is your friend from Iowa?
Yes.
Friend from Iowa.
Pharmacy?
That's me.
This is the guy that.
Do you have a pharmacy?
Yeah, they do.
You do?
Up and running?
We have.
I was about to do the pitch. So.
Excuse me. Everyone here knows who he is. He'll introduce himself anyway.
That's probably too far from
Grab your mic. So you work here at the university?
Yeah, I'm the licensing manager for biopharmaceuticals, which is a technology transfer
I
office.
know about Warfighter. When I was here, kind of PhD.
Oh, yeah,
A couple times on there.
Yeah.
One of which is with this guy.
Oh, which one?
Manganese, radio manganese for beta cells and pancreas.
It's already turned on.
John Ingalls.
I know John.
Yeah.
He's our first patient.
No. He turned his off, but yours is on, and I muted everything so you can turn it back on. Like it's just otherwise sometimes
Maybe we
I
yeah.
forget to turn it back on. Sometimes when I turn the packs off.
Yeah, then we can talk after the phone.
So you do or don't have time to go grab a beer after this?
I don't.
Birthday beer.
I mean, we can go right after.
No, right after. I don't have anything else.
The issue that he has a human, so
No,
he
I
has a
got my parents in there. Because of my.
I won't tell them. I won't tell your wife. We can have one.
Alright.
human Wi-Fi. That's why I picked one from Ukraine. So.
How long have you been for?
Since 2011, it's been a while. I used to work for Roche before. Got an MBA and got a master. I'm from Chile. Got a master in
say half of the Chileans and don't like Cubans. The other half love them.
Okay, guys. We can talk later. Do you talk to search?
I didn't park here. You don't need that. That should get you out of the garage.
Oh, cool. So I got a ticket, but I'm assuming that this one.
It came with the registration, so if you go pick up your badge, you can just have mine because I don't need it.
So, but yeah, surprisingly,
You can't do it on your own ears.
no.
Who's running it? We had two radio pharmacists. University of Kentucky still one of ours. So now we're trying to hire a second one. Of course, we have David Dick, and this is like he runs the not for so we have two radio pharmacists in Chicago. So we have we have cold chem and hot chem and which are used for that stuff. And upstairs we have cold radio pharmacy and then sterile hot compounding in the radio central lab for their therapeutic administrations.
Yes.
Yeah, so two manufacturing, one for diagnostic, one for therapy.
We've got two ladies just running the back. Andrea's just going to use the podium.
That was the issue.
Going.
Okay. Nice radiology, but I hope John helps
Oh,
out
he's
that.
better.
Yeah.
Almost like, I mean, like, he can run.
Okay, I do. Okay.
So right here,
They all know it now.
Two that are missing there also.
so what you need are GMP validated validations to go into you need somebody to write CMC sections. Hi, Steve Chris. Nice to meet you. I saw Sue and Carri downstairs, so I know they're here. Mike already. So we should, 15 minutes early to make sure I know where this room is.
I came this way, so I knew where the room was. So I came straight.
Yeah, you're in.
I was commuting from Iowa City this morning. So I'm familiar with that, so I used to live here, so I'm excited to walk down State Street over the terrace.
We're little classmates. Sort of.
Have you been over to the new short facility with Carri? In the space, right?
I haven't been to that. I think,
Is this mic on? Okay. I'm just gonna.
You said you were racing between on your teaching.
Hi. If you're seated like on the aisle, if there's open seats, can we just like move towards the walls? That way, so last minute people can just kind of sneak in. I'd appreciate it. Thank you. We'll get started once everyone's kind of settling down. This is Carri. Hello. They'll need to be Mike. Not that day.
Hello.
Hi.
Can I clip this on your lapel here real quick? And then do you have an inside pocket at all? Oh, you do not? Okay, that's fine. We can run it down. Yep.
black jacket, black shirt.
Okay. I think that's a real pocket.
Oh.
It's a real big pocket.
Careful about dating people.
I think Sue was for. Do we have more than one? Yeah.
Yeah, I'm just trying to find a place to.
Yeah, mine are like,
Yeah, I mean, usually we do like
On the back. Okay, all right, I'll do that.
Sure. Try it.
Costumes figured out.
Can you put forward your hair real quick?
Yeah.
Just bottom three if I can.
Oh, no worries.
They're actually like surprisingly easy to use.
I am like really like, oh, it'll be very nice to meet you. What brings you to this?
That's exciting.
Cable control. Just gotta double check.
Awesome.
Is it on? Well, how
Like it's on, it's muted. I have it.
So you'll cut
I
it
have control
live?
of it.
Okay, great. Oh, something here.
Sorry. Oh, sorry.
We don't see it at all either. So.
Sorry about that.
Yeah, you have to get mine.
Yeah, let me.
Last
Here,
one.
yeah. All right.
Should I have a mic?
Yep. If you're on the panel.
I am?
Yep.
Yeah.
All right.
Yeah, I'm just like an audience person. But
Okay. 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 Just coming up to talk. I get it. Yeah.
Yes.
Congrats on it. Great. I love your speech.
Well, you know, I know you've been in that blood picture. I got it yesterday, I think. Oh, yeah.
So I reviewed your script.
Oh, there's a lot, yes. There's also a lot that we can hand off to each other. And we're just gonna, don't overwhelm them with science, just make it sound cool. Yeah, my script, I'm like, is there a script? That was a strong word.
Okay, now we have a couple options on how we can do this.
On the back?
Oh yeah, yeah, I'm fine with that.
Yeah.
Let me just come onto your arm here real quick, and then I'm just gonna lift up your jacket here.
Yeah, but I mean, like, I get a. I mean, it's like a class like.
Yeah, because I can probably.
Should be good.
Can I get
Oh,
this?
yeah, yeah, I'm sorry. Yep, you're all
There we go.
Let me know what I have to do.
You want me to do like a go and
All right, I think we might as well get started. Thanks for coming in here and getting quiet so quickly. My name's Macy Camara. I'm a member engagement manager with BioForward. I'm also the one that sends out a lot of emails. So if you see your, my name pop up in your inbox, now you can put a face to the name. We've got a great session here, Going Nuclear to Fight Cancer, sponsored by BWBR. So I'm going to welcome up Andrea Cecilia. She'll say a few words and we'll get started.
Thank you, everybody. Welcome to today's panel, Going Nuclear to Fight Cancer. As Macy said, my name is Andrea Cecilia. I'm here on behalf of BWBR to sponsor this session. BWBR is a design firm specializing in complex, owner-occupied facilities with a particular focus on healthcare, education, science, and technology. With over 200 staff spread across 3 offices, including Madison, The firm has been transforming lives through exceptional environments for over 100 years. As you can imagine, we care deeply about the innovations and technological advancements coming out of your work. You are all doing such amazing things. We are honored to work with you and play a part in supporting that. From product development to fabrication, manufacturing to academia, data centers to support spaces, BWBR excels at developing high-tech environments that perform. What was once siloed environments siloed environments where scientists quietly did their research is becoming a flexible, adaptable opportunity for intellectual cross-pollination and innovation. We understand that these projects can start and stop on a dime, and once they're in motion, there is no speed but full speed ahead. As a firm with a history of valuing project management, we have the skills to manage this ebb and flow effectively, minimizing uncertainty and guiding your team through the process with grace and ease. Our team understands the complexities involved in lab or clean room design, as well as the intricacies of the regulations and codes. We know that these builds require unique integration between mechanical and electrical systems and architecture, as well as specialized expertise around hazardous materials, handling, and flow, and we can take care of it all. Understanding how people, product, and materials will flow into and out of your spaces is crucial to maximizing success and effectiveness in your projects. Our staff dig deep to truly understand the needs of all stakeholders, then leverages everything we've learned to create a plan that maximizes safety, sterility, and integrity. BWBR has worked for science and tech clients from startups to industry giants, and we provide the same level of nimble service and confidentiality to all. Our prime objective is to partner with you to support your people and your mission, both now and into the future, on projects both large and small. I'm so glad that you're here, and I know the speakers have some incredible insights. This panel of world-class leaders in theranostics will describe how they're teaming up to accelerate transitional research and clinical implementation to improve and extend lives. I don't think Sue needs any other introduction after that amazing lunch. Sue Wallace.
Yeah, did you miss me? Is that the, I think, I don't know what's going to happen on the first slide, so let's take this ride together. They talk about failing frequently. We're introducing the panel. I think come on up as you see your names, guys. We'll get up here and we'll do a little bit more formal introductions once we get everybody up here. See, these are the rule breakers. Why take the stairs when you can take the common route? Yes. Yeah, so I'm your moderator for the day, so you're on this ride with me wherever I take you, so buckle up, right? But I am super excited to be here with these three unbelievable genius brains. That's what you're going to see. And my challenge for today is how to get that essence of who they are out, to a non-theranostic audience. I want them to introduce themselves more in a minute, but I want to just say that what I'm really hoping to come out of the session today is not for you to be experts on theranostics. We'll give you one quick intro as a reminder to what that is, but to really appreciate the depth and the expertise and the complexity of the Tech Hub community we have. And we're here today as one example in the world of theranostics of the unbelievable effort and work and years and expertise it takes to do just this one thing that we do. And we've got teams all across this hub center working on different problems, equally deep, equally complex. And I hope you walk away boggled at the difficulty of what we're taking on here and really proud of what we have in this hub, in this tech center, and also hold that sacred. We need to protect that and continue to invest in that. It doesn't just happen on its own. And so I hope at the big picture, that's what we leave you with today. I really want to get to this crowd, but let me tell you what theranostics is. This is the only learning slide of the day, and then the rest you can kind of enjoy the ride. But I just wanted to put this a little bit in context. We did a theranostics session 2 years ago. People kind of said, they really enjoyed that. We talked a little bit more about the technologies behind it. Today it's more about the people and the science behind it. But the word theranostics is a combination of therapy and diagnostics. And they've combined those two words because in this field, people develop a drug and then they can use it in two ways. They can use that same drug to diagnose cancer, or they can use that same drug to treat cancer. And you're looking here at two medical images of the same patient. The image on the left is a diagnostic scan where they tagged a form of safe radiation to that drug. That drug targets prostate cancer and the metastatic disease that comes from it. So unfortunately, you see it everywhere here in the bone of this patient. That's a pretty sick person. It's not a happy story, but we can identify and see where all that disease is without harming that patient. Now we know that they're a good candidate for a therapy using that same drug, because this safe scan that I can image with the PET scanner is telling me, I know this sticks to the tumors, so if I sent it in again labeled with a toxic form of radiation, it'll go to those same tumors but kill them this time. And that's what you're seeing on the right, is now we've labeled with that toxic form of radiation. It's going to the same locations. We have to use a different imaging technology to see it, so the scan looks a little bit different, but it's the same idea. And in this case, you hope that over time it sticks there, it sits there, and it kills those tumors. So that's the essence of what we're doing in theranostics, is we're using a drug to find the disease, prove that we're going to be able to help this patient, and then we're using another form of that same drug to kill it. And they combine it to say this drug is a theranostic drug because it can do both. And that's the essence. And now I want to move on and learn a lot more about our panel and what these people do in their version of the Tech Hub, as they have different roles and they hand things off. And so I'm going to ask each of you to introduce yourselves. We'll go left to right in order here. We'll start with Reinier. But I want them to tell you briefly what they do. But more importantly, Reinier, share with us your Tech Hub story. Why the heck are you here? You know, what brought you to Wisconsin? And, you know, why is that a good thing for you, hopefully?
You got here. So thanks for that intro. I'm Reinier Hernandez. I'm an assistant professor, medical physics and radiology, and I also became recently a CTO for a local company that works on developing radiopharmaceutical agents. So my, journey was kind of interesting. I'm originally from Cuba. I did most of my undergrad preparation in Cuba, and actually radiochemistry, which is kind of ironic because there's no nuclear industry in Cuba. So, but I got a lot of like theoretical knowledge, and I knew how cyclotrons looked like and reactors, but I'd never seen one in the flesh until I came here. So I moved to the States in 2007, and I just wanted to keep on that same vein, working on the radiation field, radiochemistry. And there weren't a lot of options for grad school, and then medical physics was kind of like the most relevant. It was a little bit of a deviation from the radiochemistry chemistry perspective, but it was the only program, one of the few programs that allowed me to stay in the radiation field. So I ended up joining the medical physics department here in Madison, and then started working on with this gentleman here on developing early drugs for diagnostic and therapy. So when I finished my PhD, I had the opportunity to go many places because of the productivity that we had, but all places like look to me as a lateral or down move because the ecosystem that we have here with infrastructure, because it's a very infrastructure heavy type of like research field. So I decided to stick around for a postdoc, and then now I moved through the ranks into a assistant professor role, and I have now my lab that focuses on developing this therapeutics and imaging drugs that Sue was alluding to.
Cool. Dr. Graves.
Hello, everyone. You can hear me, I hope. So my name is Stephen Graves. I'm an assistant professor and medical physicist at the University of Iowa. I'm affiliated with departments of radiology, radiation oncology, and biomedical engineering. I started my progression through schooling and in physics in Tennessee Tech University, and spent my summers at Oak Ridge National Laboratory working on some nuclear astrophysics experiments. And I wasn't quite sure if that type of physics was what I wanted to pursue as a career. And so I would ask my more senior colleagues, if you were going to go through the path again, would you do the same thing? Would you go into pure astrophysics? And most of them said no. And so they said, you really should look into more applied physics, material science. You should think about medical physics. And around the same time, I was dating this girl in college, and her father was a radiation oncologist. And he knew I had an aptitude in physics, and radiation oncologists work with medical physicists all the time, and he sort of pushed me in that direction. But at the time, our relationship wasn't that I was taking advice from him. I said, yeah, okay, whatever, old man. But it sort of sat in my mind, and I eventually decided that was the direction I wanted to go. So I came here to Madison, the University of Wisconsin, and did a PhD in medical physics. And I got to say, I really think that the graduate program in medical physics here at the University of Wisconsin is not only the best in the US, but possibly the best in the world. And so it really is the sort of the source of a lot of intellectual property that's come about, and it is really a nice constituent of the tech hub here at the University of Wisconsin. So I wanted to pursue a formal career in theranostics, and so when I got to the point of deciding what the next step was after grad school, I wanted to go somewhere that was already doing this in the clinic. It wasn't just like they were doing preclinical research. I wanted to go somewhere where they were actually treating patients and had been doing so for more than a decade. And I only had a couple of options, so I went to the University of Iowa, where they had been doing a lot of cutting-edge research in neuroendocrine tumors and were sort of involved in a lot of clinical trials. And that's where I am now, working primarily in the translational space, taking candidate radiopharmaceuticals and trying to translate it into a practical clinical therapy that benefits patients. So I appreciate the invitation, and I work closely with Sue on a number of things, but it's an honor to be here.
All right, Dr. Glide-Hurst.
Yep. Can everyone hear me okay? So I'm Carri Glide-Hurst. I'm the Director of Radiation Oncology Physics. So I'm a board-certified medical physicist by training, so I do have a clinical practice where I support clinical medical physics activities in the radiation oncology clinic. I'm also a full tenured professor, so my tenure home is in the Department of Human Oncology, and I also have an appointment in medical physics. So I'm a Detroit native, and so I was born and raised in Michigan. And I went to University of Michigan for nuclear engineering for my bachelor's degree. Go Blue, I saw that. So my bachelor's degree was in nuclear engineering, and then I did a PhD in medical physics at Wayne State University, which is maybe not the best one in the nation, but certainly one of the oldest programs in the nation, so it was close to home. So I pursued training there. I did a postdoc for 2 years and always had a knack for research, especially in clinical trials and translational research, bringing technology to the forefront for patient care, particularly in the imaging domains. And so then I went and joined the faculty at Henry Ford Health System in Detroit, where I served for 11 years. And I established several labs and graduated many trainees over the years. And about 4 years ago, I was encouraged to apply for the big job that I have now, overseeing the physics division of the Department of Human Oncology. So at the time, I didn't think that was something that I really wanted to do. I really love my research. I love my trainees, my students, and I had a really great mentor who reached out and said, hey, you really should take a good look at this. You should come to University of Wisconsin. You should come. And I thought, oh, there's so much paperwork and HR and admin if I have to do this job. And they're like, it's worth it. Just come do it. And so I did compete for that role, and I did get that 4 years ago. And so since then, we've grown our team. We're up to 25 faculty physicists. We just launched the East Park Medical Center yesterday, so my technical team oversaw the selection and siting and commissioning of all of that major equipment. And then, so recently I've gotten plugged into theranostics, so as if there's not enough to do. The reality is you have to make these things work in the clinical setting, and so my team and my faculty really do a lot of the personalized dosimetry that you're going to hear about today. So how do we apply these to cancer patient care, and how will we, potentially personalize that for their specific treatments and tumors and their disease burden. And so we do a lot of technical benchmarking. We do personalized dosimetry for the majority of the cases, and that is really transforming how we're able to change lives. So yeah, thanks so much, and it's just a joy to be here. And I learn every day, especially from these two experts. So thank you.
Well, and I just love hearing the stories about a lot of people start in a lot of different places, but when they converge here, you see the magic. And I don't want you to miss the theme, because it's my favorite part: physicist, 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 right? Like, and we talk so much about healthcare, and I think physicians are useful. I do. But there's so much more than that behind these things that happen even every day in the clinic. And so today I'm going to ask these three to talk about what they're doing. But I'm going to hand this off to Carri and have her, it starts and ends with the patient. So she's going to help us do that and kind of have everyone else plug in what they're doing with that patient in mind.
Great, thanks so much, Sue. Okay, so exactly, everything is all about the patient and being centered on the patient. So this is a story of a patient that's well known to our clinic named Mark for today. So he's an avid traveler, so spends time in Florida. He's a snowbird. Hopefully you all know what that means. So he's a snowbird, also travels to Europe quite frequently, and loves spending time with his grandchildren, and he's very active, works out at his fitness club. But he's well known to us because he was first diagnosed with prostate cancer 13 years ago. And so we spent a lot of time trying to care for him over the years, underwent a radical surgery to remove the prostate, but unfortunately his cancer continued to progress over the years. So from there, he received a lot of years of hormonal therapies, chemotherapies, and also we can do really precise targeting to painful regions, to bony metastases, to try to keep providing him relief. And so many, many years. So again, he's been under care for 13 years for his prostate cancer. So we've really given him a lot of different treatment opportunities. Again, he tries to remain very active and also still travels a lot as much as he can. But unfortunately, last year, he was admitted to the emergency room just due to a lot of intractable pain. And they did a nuclear medicine scan. And so what we can see here, anything that's bright is essentially like a tumor burden within the bones. And so unfortunately, his prostate cancer had metastasized, which naturally goes to the bone typically. And you can see that it has a lot of burden and causing him a lot of pain. So even though we've given him a lot of therapies to prolong his life, we really, at this point has pretty widespread metastases. And so he's had so many different layers of treatment over the years, including removing the prostate hormones, and he's really failed. Actually, unfortunately, these treatments failed him, but he's gotten him also, over a decade of quality of life and continuing to live. But the question now is, what do we do with this patient, and what offerings do we have for patients like Mark in this setting, knowing that we've already thrown a lot at his care over the years, but we do want to try to do what we can to help him. And so what we're going to hear about next is how we go from like the original origins and in terms of in the lab all the way translating into the clinical care, so you can kind of understand what would it take to offer different kinds of therapy options to Mark, especially someone who's already explored so many other opportunities to treat both here in the United States and even abroad in his care. So next, I think we're handing it off to Reinier, who's going to really go back to the basics and tell us about where's the origin for everything for it starts for theranostics.
All right. So you might be wondering where these drugs come from. So, and this is kind of a busy slide, but it's intentionally because we want to kind of transmit like the complexity of like getting to these theranostics agents, and it's a multi-year process. And it takes the effort and expertise of the wide variety of types of professionals. So it all starts with identifying what makes the patient tumors different from the normal cells. In the case that Carri was alluding to, a particular protein that is expressed in the prostate cancer cells that is not expressed in the rest of the body. So that's what the first step is, identifying that target. So now what we now have to do is just identify things that will bind to that target so we can shuttle radioactive payloads to the tumor and kill those cells. So it's a long process involving a lot of resources in medicinal chemistry, tons of expertise. So we end up identifying things that will bind well to the target and spare the normal tissue. So now we have to make it radioactive. So that's where my lab comes. The radiochemistry, where we incorporate these radionuclides into these targeting molecules. But it's also where this theranostic development differs from the classical drug development pipeline, which is what we have here in the center. Now we have to rely on isotope supply and imaging complexities, and also specialized CDMOs that can handle radioactivity. So that's just to exemplify what extra complexities around theranostics we have that makes Wisconsin uniquely suited for doing this type of research. After we have made these radioactive compounds, we have to test it in animals. That's something that we do to essentially is required as part of the translational research. And then once we identify the candidates that are good, that have good tumor targeting and sparing normal tissues, then we go into the clinical testing phases. That's where professionals like Stephen here get involved. And then it goes through the normal phases of FDA testing, phase 1, phase 2, phase 3, and then the NDA. And after NDA, we have the toxicity vigilance, and that's where other off-label trials come for. So why are we looking in these drugs? So it's always a balance about efficacy and toxicity. Efficacy means like we want to maximize the amount of radiation that we deliver into this tumor, because in the end, it's a systemically administered therapy, but it's still radiation therapy. So the more radiation we give, the better response we have. But then, because the drug tends to circulate throughout the body, because it's injected intravenously, it's also hitting some of the normal tissues as it gets clear. So we have to really optimize and find the better balance, or the good balance between efficacy and normal toxicity. And the goal is to maximize the efficacy of the treatment while maintaining or improving the quality of life. In an ideal scenario, we will have all efficacy, no toxicity, but typically that doesn't happen. And I have a little bit of an example of what we do and how we try to improve some of these theranostic radiopharmaceuticals. Here we see an example of a patient that got treated with a radiopharmaceutical. All that you see in black is where the theranostic agent is getting accumulated. You radiate this. In this case, it's all tumors. You give two fractions of this radiopharmaceutical. The tumors go away. But if you notice, these salivary glands, lacrimal glands, also the uptake goes away. So that ends up limiting the impact of this particular radiopharmaceutical, because now the salivary gland, it is a toxicity that's very detrimental to the patient well-being. So in our lab, what we say, okay, let's go back to the drawing board and see if we can either improve the amount of this radiopharmaceutical that goes into the tumor, more to the tumor, or reduce the amount that goes to the salivary glands. And that's essentially how we go about it. This is the original drug. This is the one that we created in the lab, and we tweaked essentially the structure of the drug to go different places, because but basically the structure of the drug dictates where it goes, how much it goes to the tumor versus how much goes to the normal tissues. So this is tested in animals. So we see here the yellow, it means accumulation of the drug, that in the mice that receive our new version of the drug, we get much higher uptake, accumulation of the radioactivity and retention over many days. And many of these radioactive isotopes decay over weeks. So the longer this pharmaceutical stays in the tumor, the better, because we can give a lot more radiation dose. So compared to the standard drug that clears away quite fast from the animals in this case, we have a much better advantage in terms of tumor dosimetry, and then that results in these tumors don't grow as well in mice that receive our drug compared to the prior generation drug. That also leads to a better survival. So at this point, this is a drug candidate where we see, okay, this has some promise in mice. We need to move it to the next stage, and that's where my colleague Stephen here takes the baton and runs with it.
So we have a candidate radiopharmaceutical that looks like it has good data and sort of baseline observations in preclinical models that show promise. Well, translation is taking those observations and turning them into a clinical intervention that improves the health of individuals and the public. And the really cool thing about working in translational medicine is you're kind of at the nexus of not only the end user, but also the person who came up with the product in the first place. So the inventors and the principal scientists, if that has been licensed by a pharmaceutical company who's sponsoring the research, you're kind of interacting with the company and giving them advice and strategy on how to develop their product. You also interact with the Food and Drug Administration about what kind of data clinically that they want to see come out of the trials that would lead to the approval. You get a lot of insight into what kinds of toxicity they care about, what kinds of efficacy endpoints they want to see in the research. And then obviously you ultimately interact with the patients that are sort of undergoing a research study where you have to give them the informed consent, which is kind of the hallmark of clinical research, which is informing them of the potential benefits. Again, untested agents, you have to be very transparent about setting expectations with patients, and then they make an educated decision. And then ultimately, we want to come up with something that is clinically practical to implement. If you come up with something that is overly complex or requires a very specialized team to administer care, it's difficult to enable patient access. So you want to think about all these things. So to give you an idea of the process for drug delivery, I kind of wanted to take a little bit of a Disney spin. So everybody is familiar, I think, with the Balto story. This is actually Leonard Seppala and Togo, who ran the longest leg of the diphtheria antitoxin serum run to Alaska. And I think in this analogy, I'm one of the Siberian Huskies. And so the point I want to make is that at every turn in this journey of trying to bring a medicine to patients, there's opportunity for failure. There's so many ways that it can fall apart, or you didn't make the right call at the right time, and you're simply not financially sustainable. You're not able to bring the product further. And so the other thing to recognize is that while you're developing this, while you're trying to translate this into the clinic, patients are dying. Meaning the longer it takes to bring to market, the more patients will be negatively affected by that drug development timeline. So if you can make decisions that take that translational time from 7 years to 5 years, or from 8 years to 4 years, that's massive. And sometimes the FDA wants you to do that, and so they can kind of help you with that. So the kinds of things you want to think about on this path: choosing the right cancer type. It sounds obvious, meaning you have a drug that goes to prostate cancer, you have a drug that goes to pancreatic cancer. But sometimes the best cancer target, meaning the target or the cancer that has the highest receptor expression, isn't necessarily the right choice, because maybe you have other interventions in medicine already that cure 95% of them, like thyroid cancer, for example. And so maybe you want to go for the second one on the list that has no other treatment options, and there's a large number of affected patients. And so you want to be strategic at the front end about what kind of targets you want to work on. You want to choose what stage of the disease progression you're going to intervene at. Obviously, if it's a new monotherapy, usually that's kind of a last line therapy. But if you don't think it's going to work at that point, you have to start thinking about combination therapies. Patient selection. This is critical because if you have a new drug and you're targeting, say, pancreatic cancer, and so you say anybody with pancreatic cancer can be on my trial, that is a fairly non-specific selection, and it can make it so that you need many, many thousands of patients worth of data to come up with an effect that you can measure. Whereas if you had been a little bit careful with selecting the right patients for the therapy up front, like those that have particularly high uptake of your radiopharmaceutical, or patients that are likely going to live long enough to make it through the course of therapy, you're going to significantly improve the effect size that you're measuring, and therefore make the trial a lot faster, make it more likely to succeed. You want to determine the maximum safe dosage. That's usually done in a phase 1 trial. A little bit more difficult is determining the minimum effective dosage. Sometimes you have to take your best guess based on prior information, based on understanding of the radiation science. And then you balance that safety and efficacy in the target population, and you start expanding to more and more patients. You want to choose the study endpoint. So there are lots of different things that the FDA would approve a drug based on. Some of the common ones would be, how long do patients live? That's overall survival. But it could also be, how long do they live without having disease progression? That's progression-free survival. But you can be very creative. There was a radiopharmaceutical therapy called Azedra for pheochromocytoma and paraganglioma that was approved based on a reduction in antihypertensive medications, meaning it's a functional cancer that causes patients to have high blood pressure. And so this drug caused symptom relief, and it was effective for disease control, but the study endpoint was carefully chosen to receive approval. You want to do a monotherapy or a combination? That's a critical choice in something like melanoma, where we have immunotherapy that's highly effective as a monotherapy, and you might think that priming the immune system with a little bit of radiation-induced cancer killing could be a good thing. You want to think about how many patients you're going to need to do, and how many centers you're going to need to activate, and also realistically how many patients you can accrue at each of those sites, and then you start projecting timelines. You want to get FDA to bless your trial. That's obviously the first step in sort of launching a phase 2 or a phase 3 study. You identify the trial sites. And one of the problems is everybody does things a little bit differently, meaning people do even central labs or imaging studies or image interpretations or dosimetry calculations. They're all doing it differently, so you have to make sure everybody's doing things the same way. Something even as simple as measuring how much radioactivity you put in a patient, it turns out is a non-trivial exercise. You know, it's not like you're measuring a meter. It's actually radioactive disintegrations per second that you need to quantify routinely, and there's a lot of thought that goes into that kind of thing. You want to think about what kind of adverse effects are you seeing, what kind of adverse effects are acceptable. Sometimes you have to halt trials to look at follow-up. Aggregating the data and doing analysis. You have to audit sites to make sure they're complying with your protocol. Long-term follow-up with patients to look for late toxicity. Patient-reported outcomes is a new and important aspect of clinical research. If you give somebody 6 months of extra life, but their quality of life is terrible during their last 12 months, that is not necessarily a success, right? And so patient-reported outcomes are really important for being sort of soft science. How much have you improved this patient's overall disease progression? Regulatory submission is always a big one, and then FDA approval. And you might think that that's the end of the line, but it's not, because there's a difference between running a trial and trying to sell a product, meaning you have to worry about supply chain upscaling. There have been some very prominent examples of drugs that were approved, brought to market, and then they were pulled off the market because of issues in producing enough in the volume that is necessary to support the therapy. Lastly, even if you have a safe and effective drug that is approved and can be provided to clinics, that doesn't mean the healthcare reimbursers are going to pay for it. So you often will have to demonstrate that it is sufficiently beneficial relative to the cost of providing that care for CMS, for insurers to provide that reimbursement. And that can be sort of a last-minute issue that is very costly to overcome. And then finally, you want it to be sustainable. So maybe you've gone through the entire process and you found that you can benefit patients that are receiving this, but then you realize that it's a very rare cancer type and it costs a lot to maintain the drug production program. And so we've had several radiopharmaceutical therapies that have been pulled from the market because of a lack of sustainable business model. So again, this is something that people are starting to think about on the front end. And so again, any one of these issues is essentially like falling into a crack in the ice on your run trying to deliver the serum. So you want to be very careful so that you can get things to market. So what does a physicist do? Well, radioligand therapy obviously relies on interactions between radiation and the cancer cells. And we have good science on radiation biology, the effects of radiation on tissue. And the real benefit of theranostics or radioligand therapy is that you don't just have to give a fixed amount of activity to anyone. You can actually image and calculate how much radiation dose is going to the tumors and the normal organs. You can't do that with chemotherapy. Once it's in the blood, you've done your part and you have to hope it works. But with this, we can actually measure and calculate and customize the therapy. So that is to say that radiation physicists are specifically well suited to determining the maximum safe dose, the minimum effective dose, likely dose-limiting toxicities, the optimal cancer to target, so on and so forth. So there really is a role for medical physics in drug development. And I think that physicians play an important part. They're probably the Leonard Seppala that's sort of driving the sled dogs. But really it's a very broad-based team effort to bring these things to market. With that, I think I'd like to hand it over to Carri to talk about how you actually get these things going in the clinic.
Thanks so much, Stephen. So now we've seen the whole treacherous path to get there. So now let's make the assumption, of course, that we're there, and we are actually there. But the reality is it's actually pretty complicated to do this. And this is because this is truly a multidisciplinary effort. So we have nuclear medicine physicians and physicists who are involved in caring for patients for theranostics. We have radiation oncologists, who are the cancer physicians who prescribe radiation. They're involved as well. Medical physicists can be from radiation therapy or in nuclear medicine. And so we are part of the care team. We also have, as you can see here, just a whole host of things that need to go. What you heard Stephen mention is the idea of dosimetry. So this is calculating the dose or the uptake by specific organs at risk that we're trying to spare or to the tumor themselves. So when we talk about whether we're going to do personalized dosimetry for theranostics, there are some use cases where it's not performed, but let's just go through the assumption that we're going to perform it. Then we need to do our imaging and special calibration scans and have to do these calibration measurements. We have to do blood draws, all sorts of imaging and image processing that has to be done. This is mostly handled right now by our radiation oncology physicists or our support team. And then we will evaluate that personalized dosimetry into our organ at risk doses or tumor doses, and then optimize that. You can adjust the number of cycles of radiopharmaceuticals that are given. So typically it'd be like 5 or 6 cycles over the course of many months. It could be tailored down if there's a toxicity concern. It could be ramped up. So these are all part of the cycles of how to evaluate the dose and how to walk this patient through their care cycle. So one thing to keep in mind, as Stephen mentioned, is that really there's a lot of role for medical physicists. And so for those of you who aren't familiar, a medical physicist will have typically a master's or a PhD, and many of us are board certified for clinical care as well. So it's a highly technical, so most people come to us with physics degrees, engineering degrees, and from STEM fields, from really rigorous bachelor's degrees, and then into our medical physics graduate program, which really is state of the art and among the top in the world. But some other things that medical physicists do to support high precision theranostics. So we heard a lot about the developmental phase from Stephen, and the kind of the R&D phase more from Reinier. But we also have to really execute these things very well with high degree of precision. And so that really comes down to our measurement tools. So we have to characterize the machine technical performance. This is an example of our GE Star Guide that we just installed recently. And here there's all of these different detectors, and we have to determine what is the sensitivity of these detectors, what's the uniformity of the image, how do we quantify dose from these SPECT images. We use things just in time for Halloween called phantoms. And these phantoms are kind of our benchmarking physical objects that we can image. They can have radioactivity inside. And we can use that to really benchmark our quality assurance procedures on these scanners so that we make sure that what we put in is what we get out in our images. So this is a really key component for a new installation, routine quality assurance, annual quality assurance, just to make sure that when we measure a count or when we get an image out, that we trust it with high precision, because that patient's care depends on that. So we do require that really high level of care and given to this procedure. So that's very good why we have radiation oncology physicists involved, because we like to measure things within 1 or 2% and about a millimeter. So this is a perfect job for a radiation oncology physicist to get involved in. And then we heard from Stephen about this personalized dosimetry. So here we have the patient. We may have a diagnostic image. We'll give their radio-labeled uptake, and then we'll measure their uptake with a SPECT CT. Typically, we'll get a few of these scans over the course of a few hours, so that way we can measure an activity cycle. And then we'll get a real personalized dose to where that uptake is in the tumor or in the nearby organs at risk. A lot of times the organs at risk can be bone marrow, it could be kidneys. We heard a little bit about parotids or salivary glands. This is very important for swallowing function, so it's a very important toxicity to manage. And then from there we do the different cycles and then also future assessment. So along this line, we need physicists to help with the quantifying of the dose. We need to collate and register all of these images across these different time points. Sometimes we need to get really fancy with our algorithms to make sure those images match up really well. So things that are the tumor tracking over time, that's changing, we want to make sure we quantify that radiation over time and how it evolves. And then finally, we really are the technical and radiation safety experts. So patients, are radioactive and they need to be released safely. We need to manage the radiation for the personnel, make sure that it can all be executed in a safe environment for safe patient care and safe patient release. And so if you haven't heard, we're really proud that UW launched the East Park Medical Center. So we opened yesterday. This is on the east side of Madison, out by the airport. So there we launched the Radiation Oncology Clinic, where we'll be offering cancer services, which is really amazing. We had a whole day of treatments yesterday. But also very exciting is the opening of our theranostics center. So this is really a visionary strategic investment by UW Health to really focus on this destination center for clinical and clinical translational theranostics. So we are thrilled to be a part of that and to be partnering with our Nuke Med colleagues and UW Health and SMPH and the School of Medicine and Public Health, because it's bringing so much more capacity in our imaging systems and our treatment rooms and our technology offerings. So it's really, really key to the success of theranostics is keeping pace with the demand. And so we're really excited to have expanded capabilities for patients on the east side of Madison. So let's now bring everything back together and see how all of these technologies have helped Mark. So Mark underwent 5 cycles of lutetium-177. This is a radiopharmaceutical that's specifically targeted to the prostate-specific antigen on cancer cells, that PSMA on the cancer cells. Remember, this was his baseline scan. And the physicists and physicians worked very closely to estimate the dose to each of these lesions. We did more than 30 personalized dosimetry points for the volumes for those lesions. And we wanted to make sure we were very careful about the kidneys, the salivary glands, and the bone marrow, all of these organs at risk, so we can preserve function and the quality of life, like we heard from Stephen. So again, the quality of life is really so key. It's not just about prolonging life, it's about prolonging their quality of life, making sure that they continue to function over time. And here's 1 year later. So after 5 cycles of lutetium, he has 1 pending. We'll see how he does. He has a treatment response, resolution, and size reduction of all the tumors. And the other important piece is there's no evidence of new disease for this patient. So it remains stable and also just really improved. And that really, what that comes down to then is, attending more graduations for his grandchildren, more trips to Florida and to Europe, meeting 1 more grandchild and 1 more holiday, right? So these things are really important to personalized care and to personalized medicine to each patient. So it's just really an exciting time for theranostics. I'll hand it back to Sue and we'll start some discussion. Thanks.
Yeah, well, thanks. I'm so proud to introduce you to my tribe. They're so great. I just want to let that last slide sink in. That brings it home, doesn't it, for all of us? I mean, hopefully your takeaway from all the rest was freaking complicated, right? And not easy. And nothing happens quickly. These are problems. One of my many favorite quotes was from Einstein, who said, "It's not that I'm smarter than anybody else. It's that I stay with the problem longer." And that really struck me. And that's what you see each of these individuals doing, and then handing off their problem to somebody better skilled to solve the next phase of that. So I find it really inspirational. If you're having a reaction or a question, we can have some dialogue here now. So I do want to encourage you can raise your hand or step into the aisle so we see you, and we'll hear what's on your mind. Go ahead. Mm-hmm.
The use of artificial intelligence, how do you use it and in what way?
Yeah.
Can you repeat the question just so everyone hears it?
Yeah, the question was about what is the role of AI in theranostics. And I would say the role is exploding. So my research lab actually focuses on AI. And we're just now getting into this space. I think it's kind of at the early days. We have to do a lot of benchmarking, making sure we can, validate all of the work that we're doing. I think there's a real role to supplement some of the dosimetry that we're seeing. Already, this dosimetry is really high precision with these Monte Carlo algorithms that are fairly complex. So if we can simplify some of that, I think another really key role for AI here is going to be in segmenting all of the different lesions of the different, bony anatomy, and then the bone marrow. So there's a lot of organ at risk segmentation or annotations that we rely on a physician to do typically. So automating that, I think, is a key component. So we have some of that built into our pipelines, but this is a works in progress. I would love to hear, Stephen, any other thoughts on AI?
I'll admit I'm a bit of an AI Luddite in clinical practice. I appreciate the potential of AI to improve healthcare, but in terms of actual adoption, I try to incorporate things once they're mature technologies and once they've sort of demonstrated to be equivalent to what a trained expert can do. One of the examples that Carri gave was segmentation, meaning in these many, many images across time, having machine learning actually draw regions of interest around relevant objects is a clear application for AI. There are other things that people are exploring, like patient selection. So when I mentioned in the drug development process, patients are complex. There's a lot of data that goes into a patient's history, all of the procedures they've had, what their images look like. And so AI is uniquely suited to digest a vast amount of data and make a recommendation. That's one of those things that there is potential, but I'm skeptical, and I think we really need to be careful with withholding therapy from a patient because an AI-based algorithm says you shouldn't. So sort of a mixed bag at the moment, but it's certainly a rapidly growing field.
Yeah, I want to comment on that as well. So as a reminder, I run a company called Voximetry, and what we do is make software that measures that radiation and where it's going and how long it stays there, et cetera. So, and that's pretty key to them making good decisions later, that we do that well. So, so we're every day looking at what can we do within our software to use AI to make things easier. I mean, some of the examples you're hearing is they're taking sort of tedious manual work that people are doing in front of computer screens, but manually they're outlining, this looks like a tumor, I'm going to draw a circle around that and move through another, you have to do it in 3 dimensions. It can be very time consuming. We can make that more automated and faster, which is great, right? And the physicians and the physicists are like, woo, going home early, I mean, so, I think that's very well received, but I think that undersells AI, right? There's so much more we can do, and I think of meaningful AI as AI that helps us do things we cannot do. It takes us beyond the kind of individual human capability. And we're doing some things, I think, more in that vein with our software, where as an example, as we're very involved in supporting clinical trial work because they need to measure early on where is this drug going where it should and is it not going where it shouldn't, and is there so much going there that it's unsafe? And so we measure that and we can tell them you're safe or you're not safe, it's enough to kill that tumor or it's not. So we support the trials of the future drugs. And the future drugs are coming with different types of radiation tagged to it. They hope it's going to be more toxic when it gets there, but it's very hard to image. So if you can't get that picture of it, it's hard to measure. And so you say, well, we didn't see very much. I can't give you a very accurate measurement. I guess we just don't know. Okay, that's one answer. Or what we've done is used AI to drive intelligence into the technology that reconstructs those images from raw data. It's seeing these radiation events happening. And if you think of it as like it sees 5 events. So that's not enough for us to get a statistically quantitative answer. But with AI, it can say, I know enough that I can turn those 5 events into 50 events. That, based on real likelihood and probabilities and what we do know about how these drugs behave, and fill it in. So now you can get a quantitative enough answer to get a meaningful measurement, right? That just takes the measuring devices we have to the next level, right? And the other place this is going to work, and I just really want to emphasize this because we didn't draw it out too much. What's trickiest about this field is that you're all different. Like, who knew? Right? So most drugs, we say you should take 2 Advil every day, every 4 hours, and you're all the same, right? And we sort of know we're not because you take 1 and you take 4 and got it. You've self-titrated. You know that what your personalized dose is. But I think what we do know, I just am very passionate about promoting the magic bullet of radiation. What's amazing about radiation medicine is we can see what it does exactly in you. And we can measure that now, and we can see that it varies tenfold between any 2 of you, or it may vary tenfold. And we don't know based on your age, your gender, your weight, we don't know what it's going to do till we measure it in you, right? And so that degree of personalization is important and hard to predict. How do I get to the place with my software to help Carri know how much she should administer to this patient? Because what goes in your arm might be the same, but where it goes and for how long is going to be really different, and I don't know anything about you. That's where AI can help us. AI can say, I can't tell you exactly what's going to happen in this individual, but I can give you a pretty good guess if we've got data across lots of patients and we can try to find some common ground. And usually if I see these signs, it's more likely to go this direction. Or if I know this is their first cycle of treatment or their 3rd cycle of treatment, I'm going to give a different kind of prediction based on this drug, based on this phase of their disease. There's lots of layers into that AI, so it's going to take a lot of time, a lot of data, and a lot of evidence to map that out. But I think we're all playing some role in building that, and it's a really exciting vision, and only another 10 years, and we'll have some results. But I think it's really going to help us. That's where I think we're really all going together, is this personalized medicine that we can predict up front better what is the right way to treat you. We can measure if we're getting it right, by the way, after we administer it. We can still go take a look and see if we got it right for you and really drive the outcomes. But those things aren't going to happen without AI. Long answer to your question, but it's a whole nother level.
So I will add that in the front end, AI has a role. Actually, the Nobel Prize in chemistry for this year was based on AlphaFold, which is an AI machine learning. So it does help out, and we've been using AI in chemistry to predict protein structures for a while. So in that space also helps out a lot with finding the right hits and the right lead candidates for this theranostics drugs as well.
Fantastic, thank you. In terms of as this industry kind of explodes, if you kind of all hope it will and be helpful in treating patients, there's a need for irradiated materials. Do you foresee any supply issues with the irradiated materials, and how mitigating strategies to kind of secure those supplies?
Do you want to answer? Go ahead. We have an expert in the room. Yeah, you want to answer?
So I'm Adam Lynch from North Star in Beloit, and so we have a 55-acre campus exclusively. So I'm sorry, let me repeat myself. I'm Adam Lynch. Yes. Oh, sure. Yeah, it was really around irradiated materials and the supply of that source material. So what we're talking about inside of this whole progression towards the success, which we all want to see with this patient, starts and begins with that original target and what you need in order to produce that drug at the end. And so for us and for our colleagues here at Shine, it's around the production of, in this case, lutetium. For us, it's other medical radioisotopes, because in order to see this advancement occur and to start to see not only clinical trials, but commercial scale production, you need to have scale. And that scale will allow us to continue to produce more and more and be able to treat more patients. So what we want is to see not only this one case, but multiples of that. And then the other scenario that we have to talk about then is expanding clinical use and access, right? So early stage around production, thanks to what Shine is doing. What we're focused on in terms of medical radioisotope production, translating that into commercial scale so as to help more patients all over this country as well as internationally.
Adam, do you know, can you comment also on North Star as a startup? I'll call it a startup company here in the state. How many jobs have you created, would you think, at this point? You've got a lot of people down there.
Yes, we've had, right now we have about over 260 current employees, both here in Madison and in Beloit. One of our real focuses around domestic vendors and ensuring that we have domestic use and access to supplies. What we do is very specialized. This is the same case for Shine. So we're looking for, oftentimes things are just not off the shelf, but we're looking for specialized designs. We're looking for specialization as it relates to overall ongoing use and supply chains. So for us, there's really an emphasis around, as much as you can, domestic production, but at the same time you have to go out and you have to find experts in the field. And within our field of this area, Europe is still leading in this case. And so we're playing catch up, but we're doing so with a real focus around clinical treatment. Thank you.
Yeah. Great comments, thank you, and good question from Shine. And I know, okay, there's a question in the back.
Can you just do a follow-on to that? Is given the significant need, how do you scale for the patient population that's at need? I mean, how do you couple it with the supply chain, medicine, but also patient population? 4 beds is fantastic. Congratulations on the site. But where does it go from here?
So I'm going to give a generic answer, and then the experts can follow up if they want. But I do want to say, in some respects, it's not a new problem, right? The field of nuclear medicine has been around for a long time doing diagnostic work. They have found ways to scale those diagnostic forms of radiation in the most unbelievable ways. I mean, worldwide, people are being treated, you know, more than a patient every hour, depending upon the drug. You know, in cities worldwide, both impoverished as well as, you know, kind of tier 1 first-class cities. And these drugs, some of them, if you've had a PET scan, that's a very short-lived radiopharmaceutical. They didn't deliver that drug the day before for you. They delivered it 3 hours ago for you. They're driving to that hospital with little white cars 3 times a day, maybe 4, to get those drugs out to those people before they decay, right? It is unbelievable the infrastructure we have to manage and distribute radiopharmaceuticals. So in general, we got this, right? But the bigger question is, can we produce enough? For sure. And I think Shine and North Star and you know, some companies in Europe as well, all you can say is they're working on it. My impression as a novice, a bit of a novice in that field, is the question a little bit is the raw materials. You know, I think they can scale up their production. Can they get the stuff they need to create it? And I think in many cases now they're learning the science to say, we'll make our own raw materials, right? You know, you can create not radio, but you can create elements from other elements, right? If you have the right tools. And I think long term, if the demand is there, and this is really good medicine, you know, Jack Welch taught me way back when, good medicine is good business. And you know, there will be the incentives and the financial viability to figure that out. But we're actually a long way, I think, from getting there. We still have to find enough drugs that work that well, because these drugs aren't competing with nothing. They're competing with chemotherapy and immunotherapy and even radiation therapy. And the likely answers is they're going to be in combination. So how much we're going to need is actually still also a big question. Any last comments from our panel? I think we're kind of running out of time here, so I want to be respectful of that.
I think Sue was the right person to answer that, given her past at Cardinal Health. So
yeah, a little bit about that. When you're old, you've worked everywhere. Yeah, right.
Can I ask one last?
Sure.
So no one lives in a vacuum. So obviously you are not the only center that just opened up yesterday. We have that success across the country. Are you able to share information with other centers like yourself? 1 + 1 can equal 12 in this particular case?
Yeah, so I'm just going to repeat the question. Is essentially, there's lots of centers trying to solve these problems worldwide, and are they able to share information with each other to do this faster?
Right, perfect.
So I can take that one. So by virtue of moving to a center that had an established theranostics program in a world where everybody's trying to get into the game at the same time, we field a lot of questions from other clinicians at other sites, both within our state, but also, you know, the West Coast, the East Coast. So I would say over the last 5 years, I probably average 3 or 4 calls per week trying to answer questions about how do you set up clinical operations, how do you get reimbursement set up for this particular procedure, how do you prepare a room for a leaky radioactive patient, that kind of thing. So there's a lot of little details, and we write papers about some of this, but a lot of it is just having a network of healthcare professionals that feel comfortable reaching out to others that have already walked the path. That's not unique to our field. Surgeons do this all the time, right? Like you get everybody in a room and we talk about problems. Same thing goes for nuclear medicine and radiation oncology. And so that's just trying to be a good member of the community.
So I'll just add, I think on the topic of data sharing, how do we pool things for the collective good? How do we combine our outcome data and our imaging data? And the answer is it's hard. And this is what's so exciting about kind of the biotech hub and all of this, you know, influx of all this infrastructure for data sharing and to really support pooling of data across the state of Wisconsin. Nationally, there are efforts at our cooperative trial groups. NRG Oncology is our cancer oncology group. We're working to work on some prospective clinical trials. There's a lot of imaging benchmarking, standardization. So standardization's an important component of it. So I would say these are very much a works in progress. I think, you know, data handling, safety, de-identification, privacy, these are real issues. It's not unique to nuke med. It's also a problem in imaging sciences and radiation oncology. So this is really a challenge for our field. So anyone who has tools or technology to help support, this is a real unmet need that we all continue to struggle with, but also that we're all going to be working toward hopefully in this new Wisconsin Biotech Hub. So tremendous opportunity. So congratulations to the teams that put that proposal in, because it's going to be a tremendous asset for cancer patients.
Fantastic, thank you.
Thank you so much to our panel.